Hi class. This video is to talk about Chapter 5, the nervous system. But before we get started with the PowerPoint, let's go ahead and look at your Chapter 5 assignment. So here is your assignment. We can just kind of go over these questions. First, we're going to talk about homeostasis and positive and negative feedback loops in the body. Later, we'll get into action potentials. Shortly after that, we'll talk about synaptic transmission and the process of communication across the synapse. And then later, we'll get into the different branches of the nervous system. The sympathetic, parasympathetic is what you're asked about here. And then lastly, remember, this is your critical thinking where you're really putting everything into a specific concept, example. So here we're talking about the size principle of recruitment, comparing anaerobic versus aerobic exercise. So those are your questions to just keep in mind as we go through this chapter. I think nervous system has 3 parts, so let's get started with part 1. Okay, so normally I would ask, what is the purpose of the nervous system? Normally I'd let you guys sit with that for a minute, think about what is the primary purpose. So if we were to answer this question, the basic function of this system is receiving, processing, and responding to information passed to the central nervous system from our internal and our external environment. So this system plays a role in rapid communication, coordination of physiological function before, during, and after exercise. So our learning objectives, homeostasis and feedback loops will be the first thing. Next will be structure of a neuron and the conduction of impulses, which is just an action potential. We'll then get into the organization of the nervous system. That's where we talk about different branches, which you see all these branches right here. And then defining a motor unit and talking about the size principle of motor unit recruitment, getting into firing rate, wave summation of muscle recruitment. And then last and most importantly is practical applications. A practical application just means how can we apply what we've learned to a real life situation? And what we're going to do is take all the information that we've learned about the nervous system, specifically regarding exercise, and then applying it to real life. So how can we relate this lecture to the exercise that we do every single day? And that's what's most important. So let's get started with part 1. You can see what our topics are for part 1, which we just kind of went over in the objectives. But starting with basic functions of the nervous system. So first thing up here, we see responsible for conscious awareness. So conscious awareness refers to memory, our thought processes, the sensations that we feel, the perceptions that we have, anything where we are kind of readily processing information. Also reflexes, a little bit of subconscious movement involvement in there too. But basically just anything to do with when we are conscious. Voluntary control of movement. So voluntary control of movement is muscle contraction. So muscular contraction due to voluntary activation of motor units. Next is primary communication network. So this system is the basis for all communication in the body, and it works with other systems. So you may have heard of the neuromuscular system. It's how the nervous system controls the muscular system. Also neuroendocrine, which is how the nervous system regulates our endocrine system. And then neurovascular. Neurovascular is how nervous system kind of regulates our vascular system, which is chapter 6, cardiovascular system. Next, we see control of the internal environment. So talking about our internal organs, talking about nerves, and that's where we'll get into the conduction of action potentials. We see spinal reflexes. We'll talk about the spine and the main role of the spinal cord in this chapter. And then we kind of already mentioned this with conscious awareness, but assimilation of experiences necessary for memory and learning. We'll talk about parts of the brain that are involved in memory and learning. All right, so next is getting into homeostasis. So what is homeostasis? This term was first coined in 1932 at Harvard by Walter Buchanan. Homeostasis is any self-regulating process by which biological systems tend to maintain stability. This allows us to detect issues in the body and adjust to conditions that are optimal for survival. So it has to do with adapting to conditions or initiating changes in the body to remain stable or the same. We also see up here the ability of an organism to maintain equilibrium. So this system, or kind of homeostasis, we're going to detect disturbances in the environment and then elicit a change. So we think about, okay, what are some disturbances in the environment? Let's talk specifically about exercise-induced challenges. So normally what we would do in class is I would put all this up here. These are all kind of the challenges that we experience during exercise. And what I would have you do is talk to your neighbor about how, so this is what happens, all of this right here. How does the body combat these changes? And how do we elicit a change to kind of bring the body back to equilibrium? So you can pause this slide and I want you to think about this for a second because this is number 1 on your assignment. So this is important to kind of understand this process here. Okay, so increased body temperature. This one's easy. What do? Our internal temperature starts increasing rapidly. What do? We start to sweat. So we start to sweat, and that's how the body cools itself down. So that's probably a really simple one. Next, we see changes to our acid-base balance. We learned about this in chapter 2, chapter 3. So think back to that, and how does the body combat the increases in acidity that occur with exercise? And if you remember, the body has these intercellular buffers. So we're going to increase our buffering systems to buffer and clear away acidity. Next is hypohydration, which is just dehydration. Hypo meaning low hydration of the body. So what would happen here? So we're getting dehydrated because we're sweating and maybe we're not drinking water during our exercise. What does the body do? The kidneys have nephron loops and collecting ducts that if you took just your basic biology class, you would learn kind of a little bit about the kidneys. But what do those structures of the kidneys do? They hold on to water and electrolytes to prevent severe hydration. So usually when you're exercising, you have decreased urine output during the exercise to maintain the water balance in your body. Next is altered blood glucose. So the muscles are requiring glucose to produce ATP to give you energy. So you're using up your blood glucose energies, which decreases, kind of puts you close to hypoglycemia. So what happens? The body will start to break down glycogen from the muscle for extra fuel. So the body is going to pull from your glycogen stores in your muscle to give you more energy. And then let's say you stop exercising and your blood glucose levels are still low and you haven't consumed any food. what's going to happen? Now the body is going to pull from the liver. So now we're going to take the glycogen stores in the liver to replenish the blood glucose levels for the body. So that gives you a lot of information to answer the question about homeostasis during exercise and those challenges that are presented, and how the body kind of helps to get us back to equilibrium or a stable state. Now let's continue talking about that number one question, and the next part was feedback loops. So let's talk about the feedback systems in the body. So there are various types of feedback systems used for direct and indirect communication throughout the body. We have positive and negative. So this slide here looks at positive feedback loops. Positive, think, promotes or intensifies a process that is already occurring. So some examples here. Well, first we see here an action, reaction, modifier. So one just like really simple example is when we start to diet and exercise, we get stronger, we feel better, we may get compliments, which just makes us feel better, which may keep us dieting and exercising. So positive feedback loop is something that keeps going because the process keeps repeating itself. So some examples here. First example is blood clotting. So think about when you cut yourself. So if we cut ourselves, we're going to have increased blood rush to that cut or that affected area. But then platelets are going to start to surround the area, and the brain sends signals for more clotting factors, so more platelets to kind of coagulate at this area. So blood clotting is one positive feedback loop because we have to stop the bleeding. So the body sends more clotting factors to decrease the rate of bleeding and build up that scab. Next is childbirth. So childbirth also positive. Why is childbirth positive? Well, it has to do with two hormones. First hormone is oxytocin. Oxytocin increases contractions of the uterus. So when a woman is in labor, she has these contractions. Well, oxytocin stimulates those contractions and kind of increases the rate at which the body is going through that delivery or childbirth process. And the oxytocin continues to stay present throughout the entire labor process and through childbirth. And then next is prolactin, which is another hormone. This one increases milk production. So mammary gland stimulation to increase milk production, which stimulates breastfeeding, and then also maternal bonding with the child. This video right here you can watch on your own, this YouTube video. It just gives another example of a positive feedback loop, but it has to do with the environment. But let's move on to negative feedback loops. Negative feedback loops are actually more common in the body. There are more times that we have to diminish a process or reduce the intensity of a process that's occurring to get us back to homeostasis. So the positive feedback loops are actually less common. So let's talk about some negative feedback loops. So first, why do we have negative feedback loops? They're used in an attempt to return the body to an initial homeostatic state. So some examples here. This here shows sweating. So when our body temperature rises above 99 degrees Fahrenheit, the body's temperature sensors are activated. And then that sends information to the brain, which causes the release of some hormones, and then also increased blood vessel dilation, increase sweat gland stimulation to decrease the temperature, and then also increase blood flow to the skin to increase sweating. So that's how we restore homeostasis. So first one, temperature regulation, which is what I just went over. Another example is fasting. So fasting meaning not consuming food for a given period of time. So during a fast without food, we're going to have increased hunger cues at first. So at first we're going to be very hungry if we haven't eaten, but eventually our metabolism is going to slow down when we don't consume any food. This is to conserve energy. And then you also lose the sensation to eat after like 12 hours or so of fasting, or 24 hours, however long. And it's basically to conserve your energy. Metabolism slows down, so you're not expending as much energy, so you can live longer until you die from starvation. But anyway, fasting is a negative feedback loop. And then another one is blood glucose regulation. So maintenance of blood glucose is a very tightly regulated system. When we consume a meal, our blood glucose level is going to be very high, but we want to lower that blood glucose. So what happens? Insulin increases, and insulin, its job is to move glucose out of the bloodstream into the cells so they can be used for energy. So insulin increases to decrease the blood glucose level. This inhibits further insulin secretion when blood glucose now starts to decrease, and now we have regulated blood glucose levels. So again, another negative feedback loop. You can watch this video right here at the bottom. This YouTube video talks about negative feedback loops, but regards to sweating and this process up here that we just went over. All right, so switching gears, now we're moving on to neurons. So I assume that we've had a little bit of information about neurons in your A&P 1, so anatomy and physiology. So this should be review, some of this should be. So the nervous system is comprised of several types of cells with specialized structures and functions that allows the nervous system to control the body and its physiological systems. So we're going to go over just some different types of neurons that exist. The first type of neuron is a unipolar neuron. So uni meaning one. So sensory neuron with one single processed effused axon and dendrites. We see down here it's involved in touch and pain sensory information. Next is a bipolar neuron. So primarily sensory, one axon and one dendrite. So I didn't mention, but the unipolar is actually a pretty uncommon neuron, and it really only exists in the cerebellum of the brain. And we'll get to the cerebellum in a little bit, but the cerebellum has a role in touch and pain information. The bipolar neurons, these are specialized sensory neurons used for transmission of the senses. So they're involved in the pathway for smell, sight, taste, hearing, and vestibular function. So if you look up here, we see retinal neuron and olfactory neuron. So a retinal neuron of the vestibular ocular nerve, so that's cranial nerve 8, is used for sight. And then the olfactory neuron is used for smell. And then last is the multipolar. So multiple processes from one cell body, many dendrites, and a single axon. So the multipolar allows for integration of a great deal of information from other neurons due to all the dendrites they have. So if you look at all these dendrites, they have a lot more dendrites than these other types of neurons do. That's so they can receive more information. So the majority of these neurons exist in the central nervous system, so the cortex of the brain and the spinal cord. So let's look right here. You see this down here, it says motor neuron. That is the neuron that we are going to be talking about throughout the rest of this chapter. So keep in mind, multipolar motor neuron is what we're going to be discussing. Okay, so neurons, they are an excitable cell. meaning they have the ability to become excited by stimulation and generate an electrical signal. So they receive and transmit information. They're classified based on their structure and function, which we just went over on the last slide. But there are three main components and then a few other components that we'll discuss. But again, this part should probably be review. So what's the role of a dendrite, which are right here? I would normally ask this to the class, where the class would give me these answers. So dendrites, they receive inputs, so they receive external information. So that's where they receive it. And then they send it to the cell body, which is also called the soma, and it's where the nucleus exists. So it contains the nuclei with the cell's genetic information, like the DNA and RNA. And this is where the signal is processed. And then axon is the long tail-like structure that sends the impulse from one cell to the next, or from a target tissue or target cell to another neuron. So let's talk about something called this axon hillock right here. So a threshold amount of stimulus is required, which means it has to be received from the dendrites for the body, or for the cell body, to send an electrical impulse. So let's just say we have a little bit of electrical information that's received by the dendrites. It accumulates here at this axon hillock. So that's where it's going to accumulate. But the action potential does not occur until there's a threshold amount of stimulus received here. So once there's enough stimulus, it all gathers at this axon hillock, and then once we have enough, it is transmitted down the neuron, or sorry, down the axon. What is an action potential? It's a brief increase in permeability of the membrane of the cell to sodium and then to potassium. We'll learn this process on the next few slides. All right, but going a little bit deeper into the structure of a motor neuron, we already talked about the dendrites, which receive impulses and send it to the cell body, or the soma, contains its genetic material. And then this picture doesn't show it, but the axon hillock is right here. And then it's going to gather that information and it's going to send it down the axon. So sends it all the way down to a target tissue. Let's talk about something that is very important in the conduction of action potentials. So let's look here. We see an unmyelinated region, which is pointing to this little gap right here. And then a myelinated region, which is pointing to this larger area. So if we zoom in, so let's take out and we look here, what are we talking about? So we have something called myelin, and I'll just put all this up here. So we have a myelin sheath, which kind of pulls out this layer. It's a white substance high in fat, and it surrounds the axon. And then we look here and we see these little cells called Schwann cells. The Schwann cells is what produces the myelin. On the next slide, we'll talk about the purpose of the myelin. So Schwann cells right here produce all of this myelin, but then there's an unmyelinated region, which is called the node of Ranvier. The node of Ranvier is an uninsulated ion channel that we'll also talk about on the next slide. So myelin sheath, it's a white substance that covers the axon, secreted by those Schwann cells. Schwann cells are a type of neuroglia that we'll talk about in just a little bit. But let's talk about what is the role of the Schwann cells. They're involved in the process of communication by myelinated neurons. Means that the action potential hops or leaps along the axon, and that's called saltatory conduction. And that's what you can see right here. So saltatory conduction is basically where the action potential propagates into this uninsulated ion channel, and then it hops into the next one. So saltatory just means to hop or to leap. What's the benefit of this? It increases the speed at which the electrical activity, or the action potential, travels to the target cell. This allows for really rapid communication and fast reaction times. So faster connection to the muscle. And then local conduction is the process of communication by unmyelinated axons. This results in slower conduction speeds. Again, with these gaps right here, that node of Ranvier is where the action potential propagates into. Each space along the axon is about 1 to 3 micrometers. It's an enriched ion channel to which we regenerate the action potential to the next node. Increases the impulse travel velocity.
Myelin is an insulating layer that surrounds the axons of neurons. Composed primarily of lipids, myelin helps to prevent action potentials, which are the electrical signals that travel along axons, from decaying due to electrical current leaking out through the axonal membrane. Myelinated axons thus conduct action potentials more quickly and efficiently than unmyelinated axons, and because of this, many neurons in the nervous system are myelinated. Myelin does not cover an axon fully. Instead, there are intermittent gaps in the myelin where the axon is exposed to extracellular space. These gaps are called nodes of Ranvier, and the sections of myelin that are adjacent to the nodes of Ranvier are called internodes. The nodes of Ranvier are rich in sodium channels, which open in response to an action potential traveling down an axon, allowing positive sodium ions to rush in. This influx of sodium ions rejuvenates the action potential and helps to prevent it from dying out as it proceeds along the axon. Because the nodes of Ranvier are not myelinated, however, the action potential slows down at each node and speeds up as it travels along the myelinated internodes. This gives the appearance that an action potential is jumping from node to node, which is known as saltatory conduction. Myelin is formed by glial cells, but the particular type of glial cell responsible for myelinating axons is different in the peripheral and central nervous systems. In the peripheral nervous system, glial cells called Schwann cells form myelin. Each Schwann cell wraps around one segment of an axon many times to form one internode. In the central nervous system, oligodendrocytes form myelin. One oligodendrocyte can produce dozens of internodes on multiple axons. Because myelin is white, myelinated axons appear white and make up what is known as the white matter of the brain. We can see the importance of myelin to healthy neural functioning when we look at diseases where myelination is disrupted. In multiple sclerosis, for example, deficiencies in myelin disrupt neuronal communication, resulting in a variety of physical and psychiatric symptoms.
Okay, so continuing with neuroglia, which is what the video just talked about, glia cells, micro and macro. So what is neuroglia? The cells in the central nervous system are about 90% neuroglia and only 10% neurons. They don't conduct electrical signals, so they can't conduct impulses or send action potentials. Their main role is to support and nourish. So they provide nourishment and structural support for our nerve cells. So we have different types, which we see down here at the bottom. So some of them form myelin sheath across, the axons, which are our Schwann cells here, and then our oligodendrocytes. Oligodendrocytes produce myelin in the central nervous system, where Schwann cells work also in the peripheral nervous system. So I'm going to go over a few of these others here at the bottom because it's kind of interesting. Okay, so the ependymal cells, which are here at the bottom, they're involved in the production of cerebral spinal fluid, and they have a reservoir for neurogeneration after a stroke or a tumor. Next is astrocytes. These form the blood-brain barrier, which allows the provision of nutrients and maintains the extracellular ion balance, and also forms scar tissue after injury. The blood-brain barrier controls entry of substances like medication into the brain via the bloodstream and fluid. And then last is microglia, which is the first and main form of active defense of immune cells to the central nervous system and helps to regulate brain function and maintenance.
Glia is Greek for glue, and glial cells got this name because they were thought to simply hold neurons in place. We now know, however, that glia have many other functions. Glia can be divided into two classes, microglia and macroglia. Microglia act as the primary immune defense of the central nervous system. They travel throughout the brain and spinal cord and remove things like damaged neurons, pathogens, or other foreign substances. The rest of the glial cells I'll discuss are considered macroglia. Astrocytes are star-shaped glial cells with many functions, which include providing nutrient support to neurons, helping repair damaged nervous system tissue, regulating communication between neurons, and maintaining the blood-brain barrier, which keeps potentially toxic substances in the blood from entering the brain. Oligodendrocytes and Schwann cells are both responsible for covering neurons with an insulatory material called myelin. Oligodendrocytes myelinate neurons in the central nervous system, and Schwann cells myelinate neurons in the peripheral nervous system. Ependymal cells are found in the walls of the ventricles, where they produce cerebrospinal fluid, which then circulates around the brain, performing many functions, including protecting the brain from injury and removing waste products from the brain. Radial glia are involved in neurogenesis and neural development. They can give birth to new neurons and also serve as a scaffold along which new neurons can travel from their site of origin to their final destination in the brain. Satellite cells surround neurons in some parts of the peripheral nervous system, playing a protective and supportive role. Although their role is not fully understood, it is thought they might be involved with regulating the neuronal environment of some peripheral nervous system neurons.
So that 2-minute neuroscience guy is my favorite, so you'll watch a lot of those in this chapter. Okay, let's now talk about synaptic transmission. So this is kind of another question on your assignment. Talking about like, what is it? How does it work? What's this process here? So synaptic transmission, it's basically a communication process between two different neurons or neurons in a target tissue. And so that's what this says here. For the nerve impulse to continue from one neuron to another to that target tissue, it has to cross a gap, which is called the synaptic cleft. So before we get into the details of that, let's kind of talk about the different types of neurons that we have. So first we have sensory neurons, which I mentioned, which is from our senses. So it carries messages from our periphery to the central nervous system. So let's give an example of this. So senses, think of like sight, smell, hearing, touch, taste. So if we see something, that is a visual stimulus, and that's sensory. And so we send that information to our brain to where it's processed. Or if we touch something hot, or if we hear some loud noise, or if we smell something burning, those are all it's all sensory information. And so that's what's being detected here. So we're detecting the sensory information and we're sending it to the brain, which is sometimes where we have these interneurons. So interneurons are at the brain and the spinal cord. And not all processes involve an interneuron, but they can. They're connector neurons. So the main role of interneurons is to fine-tune the signal for accurate and precise motor control during a movement. So more precise response to a stimulus, meaning better execution. Interneurons are mainly involved and they're needed in sports to detect the speed of movement or location, and allows for the initiation of the desired response. So let's think about if we're playing soccer and the ball is flying right at us. So this allows the body to respond to how quickly is that ball coming at us, and how fast can we execute a motor movement to do whatever we want with it, to trap the ball, to head the ball, whatever. If we see an opponent running at us, how fast are they running? How fast do we have to run to get away from them? So kind of that's what interneurons are for, just fine-tuning the signal for execution. And then last is the motor neuron. So carries messages from the central nervous system, the brain and spinal cord, to the muscles. So this is our motor movement. So this is the actual movement of getting away from an opponent or trapping and kicking a ball. So that's where the motor part comes in. Another way to describe sensory and motor, sensory neurons are also, or signals are also called afferent. So afferent is sensory. And then motor neurons are also called efferent. EFFERENT. So just so if you see that on other slides, and if I talk about them, that's what they are. So basically, sensory information converts an external stimulus into an internal electrical impulse, an action potential. And then the motor processes that information or that signal into a desired movement.
It contains the brain and spinal cord, which is a fibrous, rope-like structure that runs through the spinal column down the middle of the back. The other part is the peripheral nervous system. It consists of thousands of nerves that connect the spinal cord to muscles and sensory receptors. The peripheral nervous system is responsible for reflexes, which help the body avoid serious injury. It's also responsible for the fight or flight response that helps protect you when you feel stress or danger. Let's examine an individual neuron up close. Here is a peripheral nerve. Each one of the nerve bundles, or fascicles, contains hundreds of individual nerve fibers. Here's an individual neuron with its dendrites, axon, and cell body. The dendrites are tree-like structures. Their job is to receive signals from other neurons and from special sensory cells that tell us about our surroundings. The cell body is the headquarters of the neuron. It contains the cell's DNA. The axon transmits signals away from the cell body to other neurons. Many neurons are insulated like pieces of electrical wire. The insulation protects them and allows their signals to move faster along the axon. Without it, signals from the brain might never reach muscle groups in the limbs. Motor neurons are responsible for voluntary control of the muscles all over the body. The operation of the nervous system depends on how well neurons communicate. For an electrical signal to travel between two neurons, it must first be converted to a chemical signal. Then it crosses a space about 1/millionth of an inch wide. The space is called a synapse. The chemical signal is called a neurotransmitter. Neurotransmitters allow the billions of neurons in the nervous system to communicate with one another. That's what makes the nervous system the body's matter.
Okay, so I hope that helped just go into a little bit more detail. So continuing, let's talk about some other kind of components of neurons. We already know they're excitable cells. So they're able to receive information, process, and send that information. Two other terms here, irritability and conductivity. So irritability causes conductivity. So we see irritability, ability to respond to a stimulus, convert it into a neural impulse. So all neurons in the central nervous system are myelinated, but not all neurons in the peripheral nervous system are. So action potentials passes through the channels that open and the sodium closes. And we'll get into that in just a second, but you can kind of see that here. And that's basically the irritability part. So their ability to convert it to a nervous impulse. And then transmission is how it transmits across the action potential. So the node of Ranvier, where the sodium channels have that chemical environment that allows for the passing of the action potential across the axon, which is conduction, after its excitation. Okay, when a neuron is at rest, there's no movement occurring, there's not sending or receiving signals. The rested state has a negative charge. So this is referred to as resting membrane potential. This results from a separation of charges, so like ions, across the neuron's membrane, and the impermeability, meaning it cannot pass through while at rest. So let's talk about permeability of plasma membranes. Permeability just means the quality of a membrane that allows chemicals, liquids, or gases to pass through. So permeability will determine when and how an action potential is conducted. Next is differences in the ion concentration. So we see over here we have potassium and chloride. Over here we have chloride and we have sodium. So that's referring to ion concentration. Each ion has its own electrochemical gradient, which serves as a driving force across the membrane, so allows it to move or to be pushed across. When the channels for the membranes open, they become more permeable. And then one more thing we want to look at here is how do we restore the separation of charges after the action potential has occurred? That's due to the sodium-potassium pump. So this pump just restores that separation of charges. We're going to talk about how each of these ions are going to flow across the membrane, so from inside to outside of the neuron, but they have to return back to equilibrium. So potassium will tend to diffuse out of the cell, which we see here from the cytoplasm to the extracellular fluid, and then sodium will move back into the cell. So it's going to move 2 potassiums in and 3 sodiums out. So 2 potassiums in for 3 sodiums. And we'll look at that more closely and we'll watch a video in just a second. All right, so resting membrane potential. No impulse is being conducted. We are at a rested state. So when the neuron is at rest, outside the neuron is mostly sodium. And that's going to have a positive charge. Inside the neuron is more potassium, and that's going to have a negative charge. Even though it says positive right here, it's talking about the overall ion concentration. It's going to be negative. So at rest, you may know this from ANP1, what is the millivolts of resting membrane potential? And that's roughly -70. So sodium ions are predominantly found outside of the cell membrane, whereas potassium ions are predominantly found inside the cell membrane. Both of these ions do have that positive charge that you see there, but there are more sodium ions than potassium ions, resulting in a negative charge, roughly about -65 to -70 millivolts on the interior of the membrane. If resting membrane potential reaches a threshold, then a nervous impulse or an action potential will occur. So what is this threshold level? It's -55, and that's in millivolts. So that's the threshold amount of stimulus needed in order for an action potential to occur. And we can see that here. We see membrane potential, and then down here we see if action potentials actually occur. All right, so we see here we have resting membrane potential, and then we see a tiny maybe change in potential. This is called a graded potential. So it's a small change in the membrane potential that is proportional to the size of the stimulus. A graded potential comes in a wide range of different sizes. So you can see this next one here. Or gradations. Gradations are kind of like a stair-stepper in force production. If just 1 or 2 channels open due to a small stimulus, such as bringing a few molecules or neurotransmitters, the graded potential may be small. While if more channels open due to a larger stimulus, it may be larger. Graded potentials don't travel very far. They just travel short distances, and they diminish as they spread, eventually disappearing. But if we have a great enough stimulus and reach this threshold, then the action potential will occur, and it can travel long distances. So across the axon, and then down to the target tissue. So let's get into depolarization and how these channels are going to open and close. So a large enough depolarization event, usually resulting from multiple depolarizing inputs occurring at the same time, can lead to the production of an action potential. So an action potential, unlike a graded potential, is an all or nothing event. It may or may not occur, but when it does, it will always be the same size. Unlike this last slide here, where we see that these different graded potentials have different sizes, but look at the action potential. It has the exact same size. Okay, so all of this begins when a depolarization increases the membrane voltage so that threshold value crosses -55. So if we're at -70 and now we're at -55, that means we're becoming more positive instead of more negative. So at this threshold, the voltage-gated sodium channels in the membrane will open. And so that's where we are on this slide right here, allowing many sodium ions to rush into the cell. So this influx of sodium ions makes the membrane potential increase very rapidly, going all the way up to about +30, sometimes even +40. So +30 to 40. And that's again in millivolts. So what are we looking at in this picture? So when looking, don't look at this bottom part, just look at this top part first. So we see depolarization. We see the exterior, we see sodium. We see that sodium is going to rush inside as that sodium channel opens. So now sodium is on the inside of the cell. So that's the change in potential. Now let's talk about repolarization. So after a short time, the sodium channels itself will close and become unresponsive to the voltage, so stopping the influx of sodium. So those will close eventually. And then a set voltage potassium channel will open, allowing potassium to rush out of the cell down the electrochemical gradient. So that's where our repolarization occurs. And we're looking at this channel now. So now that we know that potassium exists on this cell interior, well, this channel is going to open and potassium is going to rush out. So that's our repolarization. This is going to decrease the membrane potential, bringing it back towards the rested state. Okay, so after repolarization, the sodium-potassium pump that we mentioned earlier restores the separation of charge. This pump is energy dependent, meaning it requires ATP to function. So it's going to pump 3 sodium out for every 2 potassium that goes back in, so that are returned to the interior of the neuron. This process is repeated for every single action potential. So we're going to constantly keep pumping. One really interesting fact is that nearly half of all the energy consumed, so nearly half the food that we eat, is used to synthesize and maintain all of these channels and these pumps. So that's why electrolytes, sodium and potassium, are important to consume and to be hydrated. So this process continues to occur. But yeah, let's look at this picture because this kind of shows this. We see here diffusion. We know diffusion is when an ion can travel and diffuse across a membrane. So we can see some sodium and some potassium can travel via diffusion. But here's the pump. So we have to get potassium back on the inside and we have to get sodium back on the outside after the action potential occurs. So we see here 3 sodium are pumped back out for these 2 that were pumped back in. And so this restores the charge back to normal. All right, so let's go over this process kind of and put it all together because we're almost done with it. Action potential. So resting membrane potential, stimulus to depolarize. We have to reach -55 millivolts. And then we have depolarization. So this is where sodium enters into the cell, causing depolarization. We go to positive 30 millivolts. Then those sodium channels are going to close, and the potassium channel is now open. We go through repolarization. So this is where potassium moves from the interior to the outside. That channel remains open slightly longer than normal, and this is called hyperpolarization. So K channel stays open longer than needed to bring the membrane back to resting potential. This results in a phenomenon called an undershoot or hyperpolarization, where the membrane potential briefly dips lower, so to more negative than its resting potential. But eventually the voltage gated potassium channels close, and the membrane potential stabilizes at resting membrane potential. But once more becoming responsive to that voltage, the action potential cycle then begins again every single time an impulse is sent.
A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter, like serotonin or a hormone, latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction polarization which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter.
Okay, I know that was really silly, but I hope it was helpful. That's what we're going to talk about with action potentials, but we have one more topic in this video that sums up everything, kind of like what happens after the action potential once we reach the target cell. So just have a few more slides and then we'll be done with this part one. And that is talking about review of synaptic transmission. So with synaptic transmission, we already know it's a really small gap. Where are we referring to? Well, first the synapse you see is right there. We see in nanometers. It's kind of looking at the presynaptic cell from the postsynaptic cell and how do they communicate. So they're both neurons, but in order to pass a signal kind of throughout the body, it has to start somewhere. And then it is in the presynaptic cell, it crosses the synapse, and then it travels across to the next neuron and then down to wherever that target is. So here's our synapse, and let's talk about the process of communication. So upon arrival of that action potential at the presynaptic terminal, that's kind of the first part. We're going to have a release of a chemical substance from these vesicles, and we'll look at a picture in a second. So the presynaptic neuron releases neurotransmitter. That's the chemical substance. It's going to diffuse across the gap. Neurotransmitter, it's a chemical messenger, and specifically it's called acetylcholine. It's a hormone that is triggered by calcium and diffuses across that gap to the postsynaptic membrane. What is it going to do there? It's going to bind to and activate the receptor on the postsynaptic cell. So receptors are proteins designed to bind a specific substance. So it can only bind this acetylcholine. If enough substance or that chemical binds to the receptor, this causes depolarization of the next membrane, so of the postsynaptic membrane, the other cell. And then that's going to cause the channels to open, and the ions now can move across the synapse, so across that tiny gap. Then the action potential can propagate across. So here is our picture, and then we do have a few more videos to watch, and then that'll be the end. But okay, so what is this picture looking at? This is the terminal buttons. So this is the axon, and here comes that nervous impulse. So here comes the action potential, and it travels all the way down. And what is it going to do? It's going to basically activate these little vesicles. And then we see calcium over here. Calcium is going to diffuse into this area and also kind of release the chemical from the vesicle. So that's where we see here in 2, that vesicle that releases the chemical, which is a neurotransmitter, acetylcholine. And then we have the synaptic cleft, that's the little tiny gap. So this is the gap that we have to get across. So let's take this little black box here. We go up here, we see our neurotransmitter. This pink thing is the receptor. So it binds to it, which opens the sodium gate. So now sodium can flow all the way through this. And then we go to 4. So 4 is we're trying to get across the membrane. And then as it passes across, now the action potential can pass across. And then 5 is the ion closes, and now no more sodium can get through because the action potential has already propagated. Now that neurotransmitter, which is this green guy here, is going to break off and degrade.
Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused.
Okay, so that's kind of it for part 1. This video here at the top is a really great video. I'm not going to show it because it's 5 minutes and it's already been an hour, but watch it. It's really good. But yeah, that's all for part 1. Thank you for watching, and then watch the next video, which will be part 2.